I see Bob Sclafani growing on a plate. All right, well, I'm going to go ahead and get started. So before I introduce Curtis, I just want to say for any postdoc student, or really, I don't think Curtis will mind, anyone who wants to join, the hour after the talk is going to essentially be a round table with Curtis. So I know there's a couple of students that have already signed up to do it, but just basically all you do is just stay on the Zoom. And those that, when the talk ends, the rest of us will just get off it. But so particularly for trainees who want to ask Curtis about, additional questions about his talk or career advice and so forth, that's your chance. And so just hang on after the talk onto the same Zoom link. So it's really my pleasure to introduce Curtis. So Curtis was a postdoc and then an instructor in my lab until 2016. But I actually first met Curtis, it must have been, 12, 15 years ago. So I was giving a talk at Wake Forest University at the med school. And when I showed up, in the morning, I got picked up and had on my schedule that I was going to have a student host for the day. Well, guess who that student host was? So Curtis picks me up and takes me to breakfast. I won't say what kind of car he was driving, but he picked me up in that vehicle and took me to breakfast and starts peppering me with questions. Clearly had read just about every paper we'd published, and just an incredibly enthusiastic guy, just, overflowing with smarts. And so, of course, being the opportunist, I immediately said, hey, when you're looking for a postdoc, give me a call. Of course, I'm not really expecting that he would, but sure enough, 2 years later, he contacts me and ended up, of course, postdocing my lab. And so I should mention, he got his grad school, did his grad work at Wake Forest. Before that, he got his undergraduate at Florida A&M University. And at Wake Forest, he did really great work understanding how cytokines produced by dendritic cells influence CD8 T cell responses against listeria. So he really came to the lab with that sort of fundamental understanding of immunology. And his Twitter tag is even still immunology for life, if you look him up. It's really his obsession. And that was really good for my lab because it really infused that immune understanding into what we were doing and led to really fundamental changes in how we viewed aging and the aging process in terms of oncogenesis. So Curtis published, I think, 5 or 6 or 7 papers in my lab. A couple of them were really fundamentals. He published a PNAS paper and then a JCI paper that showed for the first time that selection for oncogenic events was context dependent within hematopoietic progenitor cells. And didn't just show that it was context dependent, showed the dependency of that selection on inflammation, and showed the molecular mechanism whereby an oncogenic event can be adaptive within that specific context. So really just, forged a new understanding for why aging is associated with more leukemias. He then, and by the way, he was funded by KO1 when he was in my lab. And since then, he's moved on to Emory, where he's been an assistant professor for the last 4 years. He's really gotten off to a raring start. I know he's got several papers that are about to come out. And he's really been looking at both aging, but also at obesity. And I think it's an important lesson for trainees that it's good to kind of diverge away from your previous lab's interest, and which is exactly what Curtis has done. He's really sticking more to the immune side of things, but still with an interest in cancer. And I'm going to let him take it from here, but I'll say one other thing, and that was when Curtis was on this campus, Curtis knew everybody. Everybody. In fact, I'm sure everyone on this talk probably has pressed the flesh with Curtis. He just knew everyone on campus. He was friends with everyone. I don't think Curtis has an enemy in this world.
Well, I don't know about that.
The White House at any time now. So anyway, without further ado, it's my great pleasure to introduce Dr. Curtis Henry. So Curtis, you can share your screen and take it away.
So before I begin, James, thank you for everything, not just for inviting me to give this talk, but just for also helping me grow into my scientific persona. I owe you a debt of gratitude. And to all people that are on this call that I don't know, I look forward to meeting you. And to old friends, it's a pleasure to actually interact with you again. Obviously, we wish we can do this in person. So hopefully at some point, if you guys think my work is up to snuff, I would love to come out to Colorado physically and give any updates after this talk today. But as James mentioned, I'm an assistant professor at Emory University in the Department of Pediatrics currently. I joined this department in 2016. Coming to Atlanta was basically almost coming home for me. My hometown is 4 hours away. It was a return to the South and a return to, let me see if this will work here. Let's see. Let me exit out here for whatever reason. It is, there we go. And so, yeah, it was also a return to some good Southern cuisine. So again, I live in Atlanta, Georgia. So if you happen to visit, obviously I'm friends with everyone. So feel free to let me know you're in town. We have staples in Atlanta, such as Varsity. As you can clearly see, we have these chili cheese dogs, these great onion rings that you can munch on. We have burgers. If you're in the South, we obviously are known for our barbecue here. And then an Atlanta staple is chicken and waffles. So the good part about it is, you know, if you don't have these things in excess, it's a great opportunity to really have some good Southern foods. Now, however, if you live here, this can become problematic. If you eat these diets in excess, then it actually leads to, in certain cases, an obese phenotype, which brings us to our title of today's talk, which is understanding how obesity can promote BLL pathogenesis, and also how this impacts the immune system's ability to also clear leukemia, both endogenous cells as well as in the context of immune-based therapies. Now, why did I choose to add obesity research to my research portfolio? It's very simple. I'm from South Carolina, and now I live in Georgia. And I'm also an 80s baby. So one thing I appreciated growing up in the 80s was this thing called recess. Everyone was usually very fit back in 93 to 95. This is data from the Center for Disease Control and Prevention. And what you can see is that the US in general was pretty healthy, with most states reporting less than 20% of their population being obese. Now, if you were to fast forward to 2016, we see that things they have indeed changed. If you look at the obesity epidemic, even just 4 years ago, shown here is Atlanta, down here in Georgia, and even the beautiful state of Colorado. As you see, there's no more blue on this map. Every state in the US now has an obese population that exceeds 20%. And this is a major issue both in the South and the Midwest, where in most cases, obesity exceeds 25% of the population. Now, fortunately for us, we've been collecting this data for decades. And so using algorithms, we can actually predict where the US is heading. And this is also the case for the rest of the world. And it's been shown in a very nice study by Finkelstein et al. That if we hold the current trends, that by the year 2030, so just a decade from now, it's estimated that 51% of the population will be obese, which is very, very concerning to me. Now, this is a cancer center talk, and so I'm pretty sure I'm preaching to the choir here. We know that obesity is associated with a number of diseases. I'm pretty sure everyone on this talk knows someone with one of the diseases you see highlighted here in red. But again, we're going to focus on cancer. The link between obesity and solid cancers is well established. It's been well described for diseases such as breast cancer, colon and prostate cancer. And really within the last 10 years, we started to understand that obesity is also associated with the development of hematological malignancies, with some elegant papers even coming out of Colorado by groups such as Dr. Craig Jordan's work. And so when I joined Emory, I'm in a pediatric department. So I started to research, what do we know about the relationship between obesity and pediatric heme malignancies? And so I'm just going to show you two representative examples. One is in the context of pediatric BLL. On this graph, I'm showing you survival distribution versus the time to event. We're really, really good at treating pediatric patients who are lean with this form of leukemia, such that the survival rates actually approach relatively close to 85 or 90% of patients are cured. However, if the patient happens to be obese, we see that there's a significant decrease in survival that drops almost down to about 60% of the patients receiving treatment. We're not as good at treating pediatric AML. If a child is healthy and has this disease, their survival rates over the course of 5 years is usually about 70%. But again, we see this very negative relationship with obesity, where these survival rates now drop down to about 40%. And then overall, if you aggregate just a handful of studies that are present in the literature, what you see is that whether it's pediatric BLL or AML, the survival rates typically are 30% lower compared to patients who are pediatric patients who are lean, suggesting that obesity is a negative prognostic factor for survival if any of these patients get these diseases. So one question that we wanted to ask ourselves is, why does obesity make these outcomes worse? And so the favorite hypothesis in the literature is due to the changes in the pharmacokinetics or the pharmacodynamics of the drugs. There's a lot of data to support this hypothesis. There's data showing that indeed adipocytes, or the fat cells that accumulate with obesity, can take up chemotherapies, and essentially you can't hit your target effectively because the drug never gets to the leukemia cells. Now, in science, we know that no situation is binary, and biology is always complex. And so, which brings us to essentially the working hypothesis that we have in my laboratory. This is how I used to look when I left Colorado. This is how I look now that I live in Atlanta. As we gain weight, what accumulates in our bodies is adipocytes, as shown here. But importantly, we know adipocytes also accumulating in the bone marrow, which is the site of hematopoiesis and also leukemia development. Now, adipocytes at one point were thought to be these inert cells. We know that they're not. They're illustrated here in this cartoon by these yellow cells. One of the hallmarks as an immunologist that interests me were the production of these pro-inflammatory cytokines and chemokines. And in my lab, we think that these soluble factors and co-cultures, but mainly these soluble factors, can play multiple roles to promote disease progression. We think that these soluble factors can act directly on the leukemia cells themselves, which could change both the proliferative and the survival capacity of these cells, particularly when they're exposed to drugs. But we think that an adipose-rich microenvironment could also suppress T cell activation, leading to decreases in immunosurveillance, and then an increased probability of these leukemia cells to actually escape surveillance by the immune system. And so before I go on, I will be remiss to not mention my fantastic lab. These are my current members here, and the work that I'm going to show you today has been generated predominantly by Dr. Mi-Young Lee and Dr. Anthony Ross. But again, everyone that you see in this picture have contributed to the data that I'm going to show you today, and I will try to highlight their efforts as best I can. But I also have an army of undergraduate and master students that have significantly contributed to the work that I'm going to show you today. And if you guys are online, personally, I really, really appreciate all of your efforts. Without you guys, this talk wouldn't be possible. So with that being said, one of the first things that we wanted to address in my lab was the following question. And we wanted to ask, how did obesity impact BLL progression in vivo? Again, this work was mainly conducted by Dr. Mi-Young Lee, who was actually the first person to join my laboratory. So Mi-Young and I started off to really kind of dive into this. And the way that we chose to address this question is shown here. We used the diet-induced obesity model, which is a standard model used in the field to induce obesity in mice. We chose this method because this is the way that humans become obese, is through eating foods high in fat or sugar. And so the way this model works is you feed mice, in this case C57 Black 6 mice, pellets that contain 10 kilocalories per fat diet or 60 kilocalories per fat diet. And that's showing you here after 2 months, we notice that obesity indeed is induced, at least phenotypically, in these mice. And we can see that this is the case for both male and female mice. They gain a significant amount of weight within this time period. Now, in addition to looking at weight gain as a proxy for obesity, we also look more at the physiological hallmarks of obesity, including increases in circulating insulin and glucose levels. So we performed the ELISA analysis on the serum, and we also confirmed that these mice were obese by those parameters. And so once we had evidence that we successfully induced obesity in these mice, one of the next things we wanted to do was actually to challenge the mice with leukemia cells. And so we were very fortunate to be gifted leukemia cells that harbored the oncoprotein BCR-ABL, and they also are deficient in the RF tumor suppressor. So what makes these cells so exceptional is you can actually just inject these cells into mice without conditioning. So there's no myeloablation. The microenvironment as well as the immune system is completely intact. So if you do these experiments, inject these murine BALL cells either into lean mice or obese mice, what we find is a dramatic difference in the survival of the mice. First, if you draw your attention to the percent survival of the mice fed to controlled diets, what we find is that indeed the disease is very aggressive, but you also get almost 50% of the mice that survive well over 2 months. And this is in stark contrast to mice that were fed the high fat diet or the obese mice. What we observed in this context is none of the mice actually survived past 1 month post challenge with these murine BALL cells. And so with this observation, we knew that adiposity and obesity changed the behavior of our BALL cells. So what we wanted to next do was to try to understand what were the direct impacts of adipocytes on the function of BALL cells. So the way that we addressed this question was we wanted to develop a high throughput way of actually making adipocytes and making adipocyte-secreted factors. And what I'm showing you here is a protocol that Mi Young and I found, but we modified for more consistency. I'm showing you these bone marrow stromal cells, or these OP9 cells. The stain that we use is this dye called Now Red, which stains lipid droplets. And what we can see is if you look at OP9 cells over the course of 3 days in culture, they look very stromal-like. However, if you add this adipocyte cocktail that we've created, we're able to make adipocytes very rapidly. And then by day 3 of culture, you can see that there's a lot of cells containing these lipid droplets. The morphology has changed. And so we know that phenotypically we have adipocytes. So now we can do co-culture assays or collect conditioned media and do experiments. We didn't just rely on this assay as confirmation for adiposity. We also performed biochemical analysis, and Mi Young performed a Western blot looking for fatty acid binding protein 4, which is known to be a marker of adipocytes. It's highly expressed in these populations. We see that indeed bone marrow stromal cells do express some background levels of FAT-B4, but as these cells differentiate into adipocytes, the FAT-B4 levels go up, and this is just the quantitative data shown below. Now, as I mentioned, as an immunologist, one hallmark of adiposity that I was very much interested in studying was the production of pro-inflammatory cytokines and chemokines. So just as a proof of concept, we performed an ELISA analysis looking at cytokine levels in the conditioned media. We chose both IL-6 and TNF-alpha to assay because these are two prototypical cytokines you will see found in obese humans as well as mice. And indeed, if you look in our cultures, we have significantly higher levels of both IL-6 and TNF-alpha in the adipocyte conditioned media relative to the other two conditions. And this is the color scheme that I'll try to use or stick to throughout the talk, with the adipocyte conditioned media being indicated in red, responses to stromal cell conditioned media indicated in blue, and then unconditioned media indicated in black. So now that we felt that we had a very robust system, the next question we wanted to know was, did we observe any phenotypic changes in the BALL cells cultured in any condition? So we took these human BALL cell lines here, and I'm just showing you a representative example. These are RE8 cells. We cultured these RE8 cells in unconditioned media, stromal cell conditioned media, or adipocyte conditioned media for 3 days. This is actually work performed by Jamie Hamilton in my lab. And one of the things that we observed, and we actually observed this in 8 human BALL cell lines we tested, was this pretty dramatic clustering phenotype. And to our knowledge, this isn't documented. So we're still trying to figure out the importance of these clusters. But again, the take-home message is these cells behave very different when they were cultured in adipocyte conditioned media relative to the other conditions. And then when we quantitate the average cluster size, what we noticed in every cell line we tested is that you actually get these very large clusters of leukemia cells when they're cultured in the ACM. And so we knew that these cells were changing phenotypically. And so we went from this phenotypic understanding to our desire to develop a more global understanding. And so what we decided to do next was to perform an RNA sequencing analysis of cells cultured in these different conditioned medias here, the RCH cells. And again, these are human BALL cell lines and RE8 cells. And we wanted to do this in the absence of drug. And I'm just showing you the data from our hierarchical clustering analysis. And what we see here very, very simply is that if we just focus our attention on what happens with RE8 cells, you have a very distinctive gene expression profile when these cells are grown in ACM or cultured in ACM relative to the other conditions. We see that this is a cell line specific effect at this point, but the take home message being that cells cultured in ACM induces a very distinct gene expression program that's very different from cells cultured in RPMI or SCM. Now, this was very interesting to us, so we wanted to now conduct the same experiment in the presence of a drug that's commonly used in the clinic, which is methotrexate, as I'm showing you here. This is an anti-folate drug, so it targets cells who are rapidly proliferating for cell death. And what we noticed was there was a dramatic change in the gene expression profiles when you actually culture cells in adipocyte condition media in the context of this chemotherapy. And you actually eliminated these cell line specific differences. And what we observed was that now the cells, whether they had different genetics or different biological functions, they both seemed to turn on the same gene expression program if they were cultured in ACM in the context of methotrexate, whereas if they were cultured in these other conditions, the cell line specificity remained. And so we were very intrigued by this data that two different human cell lines responded in very similar ways to this chemotherapy, which led us to our next question was this just a change at the genetic level, or was there a biological adjustment to the drug when these cells were cultured in adipocyte condition media? And so we decided to test and to look at apoptosis to various chemotherapies in multiple human BALL cell lines. And so in this slide, I'm just showing you a representative example from these RE8 cells. And I'm going to walk you through this slide because you're going to see these annexin 5 PI analysis appear pretty frequently during my talk. And so what I'm showing you here is annexin 5 PI. PI actually is an intercalculating dye that marks damaged DNA. Annexin 5 marks compromised cell membranes. So essentially anything in quadrant 1, 2, or 3 would be a dead or dying cell. So we've highlighted this using this red box, and I've also included a percentage of cells that are essentially compromised or dying. And so when we treat cells in vitro with our vehicle DMSO, we notice that over the course of 3 days, our cells are very healthy. In contrast, if you treat these cells with the chemotherapy methotrexate, again, in unconditioned media, the drugs do exactly what they're supposed to do. They kill these BALL cells. We see that there's no change when these cells are cultured in stromal cell condition media, suggesting the soluble factors present in this condition media doesn't change the ability of the drug to kill these RE8 cells. But surprisingly, if you actually do the same experiment when these cells are cultured in adipocyte condition media, what we find is that there's anywhere from a 30% to a 50% decrease in the ability of these cells to be killed by these chemotherapies. And so we actually performed this experiment several times, and we can see that that this is the case by for methotrexate, doxorubicin, and vincristine. Importantly, these are chemotherapies that are used in the clinic to treat patients. And we've seen this for RE8 cells as shown here, and with the red indicating a decreased apoptosis when the cells are exposed to methotrexate, doxorubicin, or vincristine when they're cultured in ACM. We see the same thing for a different human BALL cell line, NOM6. Again, the red lines are indicating decreased apoptosis after drug exposure. And then also when the cells, when these SCM cells are also exposed to these same chemotherapies, if they're cultured in SCM, they're more chemoresistant. So just to summarize the data from the first part of my talk, what we were able to do is create this very new system, this robust system where we can generate as many adipocytes and as much conditioned media as we would like for biological experiments. And when we actually do these experiments, if we culture human BALL cells in these adipocyte-secreted factors, we notice that there's a very dramatic phenotypic change induced by these cells clustering. And again, we can see this from 8 to 10 human cell lines that we've tested. We know that when you culture these cells in adipocyte condition media, particularly in the context of chemotherapies, we get this dramatic change in gene expression profiles that predict chemo sensitivity. And specifically when the cells are cultured in ACM, we see that every human BALL cell line we tested are resistant to multiple classes of chemotherapies that are used to treat patients. And so this data got us really, really excited. And one of the things that we really wanted to do was to understand if there were any genes in our RNA sequencing analysis that would help us identify proteins that would promote both cellular adhesion and then also chemoresistance. So in theory, these proteins would have to be involved in have both adhesive and signal transduction properties. And so what we decided to do, and I'm going to summarize several years of work within one slide. And I always love when people do that. That means that you absolutely love what you do if you can distill 2 years of your life down to one slide. And so I'm going to attempt to do that now. Our criteria for analyzing candidate proteins, where we were looking for genes that were significantly increased in our RNA sequencing analysis when cells were cultured in adipocyte condition media relative to our other conditions. Specifically, again, we were looking for genes that coded for proteins that had both adhesive and signaling attributes. And with that being said, we narrowed our list down to 20 candidate proteins that would be expressed on the surface of BALL cells. And we wanted to test if these proteins were upregulated when these cells were cultured in our adipocyte condition media. And importantly, we were looking for proteins that showed a consistent upregulation across 10 human BALL cell lines. And to summarize, the protein that tracked the most with these criteria was actually Galectin-9. And when we found this, my first question was, what is that? And so that's what we're going to address next, because I'm certain that many of you may not be aware of this, what I find to be a very cool protein and very cool lectin, Galectin-9. And so there are actually 15 members of the Galectin family that's been discovered in mammals, some expressed exclusively in birds, some in fish, and some in humans. But specifically, Galectin-9 is a small member, a small lectin of this family. It binds to carbohydrates that expressed as beta-galactoside moiety. It actually is a biphasic protein, meaning that it's connected by a linker, and both terminal have a carbohydrate recognition domain that can recognize both N and C proteins that are glycosylated. In terms of ligands, Gal9 has been known to interact with TIM3, Selec7A, CD40, and CD137, with this interaction with TIM3 actually being the most characterized. And regarding its interaction with TIM3, the Gal9-TIM3 interaction actually has been shown to be immunosuppressive. When Gal9 binds to TIM3 expressed on activated or effector T cells, what it does is it reduces proliferation of T cells, it reduces cytokine production, cytolysis, and at high levels, it actually can promote apoptosis in T cells. And then more recently, and I'll get into this in a few slides, there's been shown in the context of acute myeloid leukemia that Gal9 can actually feed back on myeloid cells or myeloid leukemia cells and actually promote their stemness. But again, I'll go over that in a few slides from now. Now, the one thing I do want to highlight, which are two papers, actually one paper, but these papers were published in tandem in Nature Communications in 2018, which really helped me understand and get excited about Galectin-9. And I'll just kind of quickly highlight this. This is one of the papers that I found to be very important for the research that we're doing in my laboratory. And this is showing you the way Gal9 works on normal B cells. So I'll try my best to walk you through this. And so on the legend here, if you pay attention, this little figure here represents endogenous Gal9. You have the B cell receptor, which would give you activating kinases to your cells. And then you have these inhibitory receptors that usually have phosphatases associated with their cytoplasmic domain. And so what happens on the surface of a B cell, and this paper did a fantastic job of showing this, is that if you have Galectin-9, if it gets trafficked to the surface, and here's your B cell receptor again, what will happen is you will ligate your B cell receptor with this inhibitory molecule, CD45 or CD22. And overall, if these B cells are trying to be stimulated by ligating the inhibitory and activating receptor, due to this phosphatase domain, you actually attenuate normal cellular signaling. And so basically, in a nutshell, the way to think about this is Gal9 actually acts as a brake to slow down cells from hyperproliferating. And so this was a very important observation that they made in this paper. And I really think that it allowed us to move forward with this project. And so Even though I didn't know a lot about Gal-9, I was very pleased to see that people are starting to pay attention to Gal-9. So the literature is not very dense, but it's very convincing. In the context of solid tumors, what we actually know about Gal-9 is it's associated with the pathogenesis of breast cancer, and then also nasopharyngeal carcinoma. Gal-9 has been shown to actually promote the induction of MDSCs, and creating this pro-inflammatory environment that helps these cancer cells proliferate. And then also in this study, they showed that patients with high circulating Gal-9 levels actually have higher mortality rates. So in theory, circulating Gal-9 levels could also be used as a biomarker, at least for certain diseases. But as I mentioned, in the context of heme malignancies, the most of what we know about Gal-9, which isn't a lot, it falls into our studies in acute myeloid leukemia. This is actually one of my favorite papers on this topic. It was published in Cell Stem Cell about 5 years ago. But what it shows is that Gal-9 can be released from AML cells, bind to TIM3 expressed on the surface, and you get the activation of several intracellular cascades, most notably beta-catenin, which helps maintain these leukemia stem cell signatures and maintain stemness of these cells. So this was a very important paper for us as well. And then in addition, there's other studies in AML that shows that TIM3 is actually important for trafficking Gal-9 to the cell surface. And as I mentioned, it shows that AML cells can actually release Gal-9. It binds to TIM3 on activated T cells and actually promotes immune escape. And so the literature is starting to proliferate, but there's nothing in the literature involving Gal-9 and BLL pathogenesis, and particularly in the context of obesity. And so we hope to be able to contribute to this body of literature, hopefully very soon. And so, and the reason why we feel that we can contribute, I'm going to show you a few slides here that hopefully you find interesting. We next asked the question, what role does Gal-9 play in BLL pathogenesis? And again, specifically in the context of obesity. And so one of the first things we did was we went back to our human BALL cell lines. We cultured our cell lines in these condition media shown here with the same color schemes as I alluded to earlier. This is qPCR data looking at these fold changes relative to actin. And what you can clearly see is if you look at the human nomenclature for Gal-9, this is L-Gal-S9, we see significant increases in these gene expression profiles in all of the human BALL cell lines we tested. Now, in addition to increasing the gene expression profiles, if you actually culture these cells in a adipocyte condition media and then look at the surface expression of Gal-9 using flow cytometry, what we find is that the Gal-9 surface expression is very low when the cells are cultured in RPMI or SEM. You see that there's no difference compared to this isotype control staining. However, when the cells are cultured in ACM, we see that you do get a significant upregulation or induction of Gal-9 surface expression. And we've seen this over multiple cell lines showing you Gal-9 MFI to varying extents, but all significant in every case tested. Now, in addition to looking at human BLL cell lines, we wanted to know, were there any relationships with the human BLL, primary human BLL cells themselves? So we went to the St. Jude's Pecan data portal. Jamie did some data mining for us, and what we wanted to assay was, does the expression of Gal-9, or is it increased in different human BLL subsets relative to levels found in normal human PBMCs? And as you can quickly see, the answer was yes. In most cases, the Gal-9 gene is more highly expressed in various BLL subsets compared to normal PBMCs. We worked with Dr. Christopher Porter, and we actually performed more data analysis looking at the target data set from the NCI. And what I'm showing you here is a survival probability over years. And this is in the relapse context of BALL. And what we find, if you actually mine this data, is that patients with high levels of Gal-9 actually have poorer survival outcomes compared to relapse patients that express lower levels of Gal-9, suggesting that, again, Gal-9 is highly expressed in different leukemia subtypes, BLL subtypes to be exact, and that high expression actually leads to, or could lead to a more aggressive disease. Now, one of the benefits I have for being in this department is that Aflac has provided substantial funding to create multiple biorepositories. And so every patient that we see here at Emory, particularly the leukemia and lymphoma patients, we actually get samples from these patients, peripheral blood, serum, bone marrow, etc. And so one of the questions we wanted to ask with this valuable resource is if we actually stain for the surface expression of Gal-9 on BALL cells isolated from lean versus obese patients, did we see a difference? And so for the sake of ease, I drew this arbitrary red line to kind of draw your attention to the fact that BALL cells from lean patients actually do express surface Gal-9. But however, if you look at the expression from obese patients, we see that there's, in general, a higher percentage of cells that are now Gal-9 positive, and you see a lot more cells expressing very high levels. And we actually have done this for numerous patients, and that data is quantitative here. So if you look at the average surface expression of Gal-9, you see that is indeed higher on BALL cells isolated from obese pediatric patients with this disease. Now, in addition, we wanted to look at the data in a different fashion, kind of a more granular fashion. And so what we decided to do was look at the Gal-9 surface expression on BALL cells relative to the BMI of the patient in which the samples were obtained from. And what we found was a very strong R-squared value, suggesting a very strong relationship, almost approaching 1, whereby patients with the highest BMIs actually had the highest levels of Gal-9 expressed on the surfaces of their cells. So given that we had convinced ourselves using multiple ways that Gal-9 indeed tracked with adiposity and high BMI, the next thing that we wanted to do was actually to block Gal-9 to see what would happen to these cells. And fortunately for us, there's actually a commercially available antibody, this 9M1G clone that's very specific for Gal-9. So we purchased this antibody, and we initially sought to test the impact of using this anti-Gal-9 blocking antibody on cell cycle progression. And that's the data that I'm showing you here, looking at EDU versus PI analysis. And so this data will allow you to look at cells in sub-0 Z1, which are usually either quiescent cells or dead cells. We have G0 to G1, S phase, and then G2M. And for the sake of these, I put the percentages here in red for the cells in G2M because that's where we saw the biggest effect. And so what we notice is if you don't add anti-Gal-9 antibodies to these cultures and you allow BALL cells to be cultured in this conditioned media for 3 days, we see that there's a similar percentage of cells in G2M. However, if you add this anti-Gal-9 blocking antibody and look over the same time period, what we notice is that there's a dramatic number of cells that accumulate into the G2M phase, but you also see a little bit of accumulation in the cells in sub-G0, suggesting that, blocking this lectin may lead to deregulated cell cycle progression. And that data is just shown here quantitatively, just showing you this pretty dramatic increase in the G2M phase of the cell cycle. And so what Mi Young and I decided to do at this point was she performed Western blot analysis to look at certain cell cycle regulators to see if they were deregulated when you added the anti-Gal-9 blocking antibody. And so this is the data that we found. And so again, at baseline, we don't see a lot of difference when you look at cyclin D3 or cyclin CDK4 or gamma H2X, which is a proxy for DNA damage. However, in adipocyte conditioned media relative to results found in unconditioned media, stromal conditioned media, when you add the anti-Gal-9 antibody to these cultures, you feel that you completely ablate cyclin D3, you completely ablate CDK4, and you also get this concurrent increase in gamma H2X, suggesting that this deregulation is associated with more DNA damage in these human BALL cells. And that's just reiterated here. This is just the quantitative data from multiple experiments. And again, the take-home message is that if you add this anti-Gal-9 antibody in a adipocyte conditioned media, you get rid of CD3, CDK4, and you get more DNA damage. And we were really, really interested in the DNA damage component of this response. And so we decided to take this a step further. And the next question we asked was, do you actually see an increase in activated caspase-3 or cleaved caspase-3? And so this experiment was very informative, because what we found was even just culturing our cells in a adipocyte conditioned media led to some cleaved caspase-3 activation. So we think that cells become a little bit primed in this condition. But importantly, if the cells are now exposed to the anti-Gal-9 antibody treatment, you actually now can induce a little bit of DNA damage apoptosis in cells when they're cultured in unconditioned media or stromal cell conditioned media, but you dramatically induce cleaved caspase-3 activation when cells are cultured in this adipocyte conditioned media. Now, Again, this is cleaved caspase 3, suggesting apoptosis. So we went back to our assay to look at apoptosis, again, the annexin 5 PI analysis. And so what we found in these cases was with the amount of apoptosis or dead or compromised cells indicated in these red boxes, is that if you add anti-Gal9 antibody, and I'm showing you the results for 2 human BALL cell lines, you get a little bit of apoptosis in both of these cell lines with the single agent anti-Gal9 antibody treatment. In stromal cell condition media, you also just get a little bit of apoptosis, but in adipocyte condition media, and this has actually been consistent for 10 human cell lines over at least 7 experiments per cell line, what we find is that you get anywhere between 80 and 100% cell death, and that's just shown here quantitatively. And so we were extremely excited about these findings, but we decided to take this a step further because all of these studies that we have shown you so far have been in vitro. And so we wanted to next ask the question, if you allowed obese mice to be treated with this anti-Gal9 antibody, did it have any effect? And so the experimental protocol that we used is shown here, and I'll walk you through this. So again, in this experiment, we had lean and obese mice, and we actually, in this experiment, use either the murine BALL cells that I alluded to earlier, or one of our human BALL cell lines. So we did xenograft studies. We allowed the leukemia to actually manifest in the mice. So when the mice exhibited signs of morbidity, and this occurred usually by day 7, the mice received the following treatment. They were treated with vehicle, either single agent methotrexate, single agent anti-Gal9 antibody, or the combination of both. And then we monitor survival. And then for the sake of today's talk, I'm indicating significance using this red check mark. And so I'll walk you through this data because there's a good bit of data here. And so when we look at the impact of these treatments in wild type mice fed a controlled diet, this red asterisk indicates when the treatment began. Mice receiving vehicle, as I showed you earlier in the talk, these mice are very susceptible, and we actually get 100% cell death within a month. However, if the mice were treated with methotrexate, we see that there's a significant percentage of the mice that survive, and the anti-Gal9 antibody is less effective, but kind of effective in these controlled mice. And so when we actually did statistics, the only thing that actually turned out to be significant was mice treated with methotrexate. The purple line is actually superimposable with the green line, suggesting that the anti-Gal9 is not acting synergistically with the methotrexate to extend the survival of mice. Now, what became really interesting to us is when we actually performed these same treatments in mice fed to high-fat diets or the obese mice, what we find is that the methotrexate treatment is actually very ineffective in obese mice, whereas the anti-Gal9 antibody treatment is extremely effective, and it actually led to the best responses that we saw in these experiments. And when we did the statistics, what we find is, again, there's no synergism between anti-Gal9 and methotrexate treatment, suggesting that in obese mice, most of the effects that we were observing in this experiment was due to the anti-Gal9 antibody treatment. And so we conducted these experiments also in immunodeficient mice, NOG mice to be specific. Similar to what we observed in wild type mice, we found that the methotrexate gave us the superior response. And again, no additive effect when you added Gal9 to this backbone. And then the obese mice also gave us the same effect, or the obese immunodeficient mice also gave us the same effect. The anti-Gal9 antibody treatment indicated here in orange gave us superior results compared to the mice that received the dual treatment. Again, no difference. And methotrexate was less effective in obese immunocompromised mice. But the biggest difference that we saw between the mouse strains is that none of the mice survived treatment, suggesting that the adaptive immune system does play an important role, but anti-Gal9 also can be effective even without the adaptive immune system being present for part of the time. So just to summarize the second part of my talk, what we found is that in the context of adiposity, we feel like you get this priming scenario where these adipocytes are secreting all of these soluble factors around. This is leading to increases in DNA damage. In data that I'm not showing you today, you get a slight decrease in proliferation, but as a response to this microenvironment, both human cells, primary cells, they all upregulate Gal9. And we think this response was to protect the cells from apoptosis. And indeed, when we add single agent and anti-Gal9 antibody treatment, we feel like we get this execution phase where now these cells dramatically upregulate DNA damage and cleave caspase 3 activation. You get this aberrant cell cycle progression and a ton of apoptosis. And we can see that this single agent antibody treatment actually protects obese mice from BLL pathogenesis. So we're very excited about the story, and hopefully at some point we can actually see the story in print very soon. Now, as I mentioned in previously, what we did also observe from those experiments, the in vivo experiments, is that in immunocompromised mice, you weren't able to completely cure the mice if the mice didn't have an adaptive immune system. And this plays into the second area of research in the lab, and the second story I'm going to show you today, which is what were the indirect effects of adiposity, specifically on the T cells' ability to control BLL. And so here's our little friend of 2020, COVID-19, right? So you can't get away from it, and this presentation will be no different. So in the infectious disease world, it's been well established that obesity actually leads to compromised immunity or altered immunity. And this is actually a paper that, as you see, was published at the height of the pandemic. And we also have found that in terms of COVID-19, obese patients were more susceptible. And this was actually attributed to deregulated inflammation, poor T cell responses. There may be issues with vaccines, but I guess we'll find out next year. And overall, due to these defective T cell responses, there's been in several different disease contexts, and this may be the case for COVID-19, that the T cells have a harder time actually surveying for infected cells. And so in the ID world, the relationship between altered immunity and then poor outcomes is well established, but not as much in the context of heme malignancies. And so we really wanted to try to understand if obesity altered the ability of T cells to survey for leukemia cells and kill these cells. And so this story was actually focused on by Dr. Anthony Ross, who is a pediatric oncology clinical fellow in my lab. And so I'm going to summarize our mice data and then really focus on our human data in these last few minutes. And so I may be dating myself. Here's the Cliff Notes version of our murine studies. What Mi-Young did in assistance with Anthony was we actually isolated T cells from lean and obese mice, and we performed qPCR analysis. And in summary, what we found was that if you look at the expression of genes that code for TCR machinery, they were all down. And we noticed this in both CD4 as well as CD8 positive T cells. Now, Anthony was able to take primary T cells from mice. And what he found was that if you did co-culture experiments with adipocytes or activated these cells in the context of adipocyte conditioned media, and he used various forms of stimulation, what we found is that these T cells significantly upregulated PD-1. There was decreased interferon gamma and TNF-alpha production, and then also attenuated production of cytolytic mediators, specifically preferrin and granzyme B. And so we had all of this evidence that these murine T cells were defective. And so we wanted to go back to our biorepository and ask, do we see the same thing if we actually isolate T cells from patients with leukemia? And so we purified T cells from lean patients with B-cell acute lymphoblastic leukemia. We stimulated these cells, and I'm showing you the primary results from on day 3 post-stim. And if you look at interferon gamma indicated here in this red box, we see that 14% of the cells are capable of turning on or making interferon gamma, and that 12% of the T cells can also make TNF-alpha. So you do have some functional T cells from patients with disease. Now, if these patients are obese, we see that interferon gamma production is not altered, but you have a significantly blunted TNF-alpha response. So these cells can't turn on TNF-alpha. And so this is just showing you, shown here quantitatively, no difference in interferon gamma production, but we notice from patients that endogenous T cells are defective in their ability to make TNF-alpha. And so given these compromised endogenous responses, we actually reached out to Dr. Priscilla Doe, who's a postdoctoral fellow in my colleagues Eric Drayden's lab. And Priscilla performed the following experiment with endogenous T cells, in this case taken from healthy people. And what Priscilla did was she isolated these T cells, and these T cells were labeled with the cell proliferation dye. She co-cultured these T cells with labeled leukemia cells in the context of this immunotherapy blinatumomab. And blinatumomab is actually FDA approved to treat relapsed and refractory patients with BLL. So this is an immunotherapy that's actually used in the clinic. And so Priscilla performed this experiment in the condition media shown here. And what she found was after 3 days of culture, the cells that were cultured, or the T cells that were cultured in unconditioned media, or stromal cell conditioned media, proliferated, and that's shown here by the peaks moving to the left on this slide, whereas the T cells cultured in ACM did not. And when she looked at cytolysis of the labeled leukemia cells, if you focus on the conditions where blinatumomab was actually present, over the course of 3 days, we can see that unconditioned media and stromal cell conditioned media give you very substantial killing of the leukemia cells. And this is not the case for cells when these experiments were performed in adipocyte conditioned media. And then here's the quantitative data showing you that over the course of 3 days, you get very substantial killing of the leukemia cells when you perform these experiments in unconditioned media, stromal cell conditioned media, but this is significantly reduced when these experiments are performed in adipocyte conditioned media. And so I don't have to tell you guys about CAR T cells, so I won't spend a lot of time on this slide, except for to say that CAR T cells essentially are endogenous T cells engineered to see their to see their targets. And so Anthony and I actually postulated that there may be issues if you take defective T cells out and try to engineer better killers. And so we think that you may not be able to do this effectively. And then also the second issue that may arise is you're taking these engineered CAR T cells and you putting these engineered CAR T cells back into hostile territory, again, patients with chronic inflammation. And so we feel that in obese and maybe even in aged patients, that there may be defects here and maybe rooms to actually try to modulate the responses of these T cells to give you better outcomes. And so in the context of obesity, we wanted to look at how CAR T cells, and these are CD19 directed CAR T cells, behaved when we cultured these cells in adipocyte conditioned media relative to the other conditions. And I was fortunate enough to work with a very talented clinician scientist, Dr. Sunil Riker, who's a pediatric hematologist oncologist here at Emory, and he's also a CAR T cell expert. And so when we actually got these CD19 directed CAR T cells, Anthony decided to first look at PD-1 as shown in the box, and unstimulated and stimulated CAR T cells. What we found was very interesting. When you just put these cells in adipocyte conditioned media, we see that there's a differential regulation of PD-1, but as expected, PD-1 surface expression is upregulated when the cells are stimulated, and that's just shown here. Again, this aberrant increase in PD-1 expression on the surface of these cells, and this is an immunoinhibitory protein, and then an increase of this protein when the cells are stimulated. Now, when we look at TNF-alpha production here along the x-axis and shown in these red boxes, unstimulated cells don't make TNF-alpha as expected. And CAR T cells, when you stimulate these T cells, we see that these T cells are very capable of being or turning on TNF-alpha production unless you actually try to stimulate these cells in adipocyte conditioned media. Again, TNF-alpha levels are significantly lower than what we see in our control conditions. So in addition to looking at cytokine production, we wanted to look at cytolysis of our labeled leukemia cells. And so Anthony performed a similar experiment to what Priscilla performed, and with the major difference of we wanted to look earlier at 4 hours. And what we were able to find is that in RPMI and stromal cell conditioned media, these engineered T cells, or these CAR T cells, actually are very potent killers. Even in a short time period, you're getting almost 40% of the cells of the leukemia cells being killed. However, when the cells are cultured in adipocyte conditioned media, we actually see that none of the cells die. We've actually done longer time periods, and you never actually get over 50% cell death. And so we're very confident that at least exposing these engineered CAR T cells to adipocyte conditioned media may compromise their function. And so we wanted to look at this a little bit deeper and really try to understand, is it just the CAR T cells or just the endogenous T cells, or are the ALL cells changing themselves, at least in terms of immunogenicity? And so this was an experiment that was performed by Aida and Jamie. And what they did was we just simply cultured our human BALL cells in the conditioned medias and shown here. And we looked at the expression of CD19 on the surface of these cells. Again, this is the target for the CARs as well as blinatumomab. And then we look for the expression of two immunosuppressive mediators, both PD-L1 and PD-L2. And the business end actually happened at 48 hours. What we found is you actually have this dramatic downregulation of CD19. So the antigen that we're targeting is actually downregulated. PD-L1 goes down, is downregulated, but you get this compensatory increase in PD-L2. And we've seen this over multiple experiments suggesting that the leukemia cells are actually changing not only their antigen expression, but also their immunogenicity, and may be able to more efficiently attenuate T cell responses. So just to summarize this last part of my talk for today, what we found from our murine studies is shown here. If you isolate T cells from obese mice, we find that there's decreased expression of genes involved in TCR signal transduction, defective proliferation. We also observed attenuated cytokine production and a failure to upregulate cytolytic machinery. If you do these experiments using human samples, we see the same effect, most notably a decrease in the killing efficacy of two FDA-approved immunotherapies and a downregulation of CD19, the antigen that we're targeting, and subsequent upregulation of these immunosuppressive mediators. And so we have, in addition to what I showed you today, several things in the pipeline. Young is actually studying, and hopefully we will follow up with our BLL paper with the TLL paper, looking at the role of Gal9 and TLL pathogenesis. And we have another study where we've done omics pretty much on every cell line and primary patient sample that we have. And we look forward to trying to put out an omics paper on this topic next year as well. Jamie is looking at how elevated levels of IL-9 in the obese microenvironment actually promote BLL pathogenesis. He has an ASH fellowship on this and won several awards, so we're excited about this project. And then Aida is the newest member of my lab. She's looking at the relationship between Gal9 and DLBCL development, so this is lymphoma. And then we also received a provisional patent to make Galectin-9 directed CAR T cells. We have about 5 constructs that we're in the process of testing. So Aida will hopefully be able to report back on that soon. And then last but not least, I'm working with several groups at Georgia Tech using engineering approaches to look at how, and this is work done primarily with Khalid Salida and his very talented postdoc, Miss Anna Kellner, looking at TCR biomechanics in these various conditions. I'd like to thank everyone at Emory who I've worked with, all of my funding sources, and then also my beautiful family. We want to send you happy holiday wishes since Christmas is rapidly approaching. This is my wife and this is my 2-month-old son. So this is a day and this is Chidi. So later on this afternoon, I get the privilege of going home and playing with both of them. And then this is, again, my lab family. This is BC or before COVID, as you can see without the mask. And so from both of my families to yours, I want to say thank you guys for having me today. And I look forward to taking any questions that we can squeeze in the time that we have left. Thank you.
That was really fantastic, Curtis. That was, I mean, really important stuff. So kudos. So I'll start off. Well, you know what? I'm gonna save my question because you and I can chat at another time because you got a bunch of questions in the queue, and I'll let everyone else ask. So I'll just go in order. Steve Nordine's got the first one, and if you're asking a question, go ahead and turn on your camera too, if you don't mind, so we can everyone can see happy faces.
Well, James, my camera is on, but my computer has a glitch.
Oh, okay, no worries. But you want to just ask your question then?
Lovely face.
Hey, Steve, how are you, man?
Very simple question, Curtis. How about PARP inhibitors, synergy with the anti-Gal9?
So yeah, so that's a great question. So just to repeat Steve's question, he asked, will the ERK inhibitors actually synergize with anti-Gal9 antibody treatment? The short answer is we don't know yet. We haven't done a lot of the inhibitor treatments. We focus mainly on chemotherapies, but that is certainly something that we can look at. I'm certain Jamie is online somewhere. One of my students is online taking notes. And so we'll certainly add that onto our to-do list for 2021. So yeah, that's a great question. Thank you, Steve.
Yeah, that's cool with the PARP inhibitors. So so next question is from Paul McLean.
Hey, Curtis, great talk. That was fantastic, and congratulations to the new little one that you have in the house.
Yes, thank you.
My question is on your conditioned medium. You know, in the adipocyte biology world, there's mesenchymal-derived adipocytes and myeloid-derived adipocytes, and you're using a myeloid-derived cell line to prep your conditioned medium. And those are supposed to produce really bad adipocytes, pro-inflammatory. So 2 questions real quick on that. Have you tried any other different cell lines with your conditioned medium, or have you followed up with studies to say what is in that conditioned medium that's doing the business?
So I think you must have been a reviewer of pretty much every grant or every paper I've submitted in the last 2 years. So we are actively trying to identify the active ingredient. Matter of fact, and that goes back into the omics paper I was talking about. We've done mass spec on 20 patient samples compared to 20 non-disease samples. I actually get that data back this week. So I have a friend who actually did his PhD at Colorado. And so he's now a proteomics person at this company called Shimatsu. So if anyone knows Ryan Walsh, so Ryan and I are still buddies, and they actually did these samples for free. And so I will know what's actually circulating in the patient soon. We actually have done proteomics on the condition media and Luminex analysis. To make a long story short, that's why we started looking at IL-9 a little bit, and we see that it can drive disease progression. It actually induces STAT3 and ERK. And we actually have this interesting finding that it may actually reduce methotrexate uptake. So we have labeled chemotherapy, and we can do live cell imaging. And so we're starting to hopefully deconvolute what's in the soluble microenvironment to see what impact it has on these cells. And regarding your second question, other cell lines, like I mentioned, AIDA is looking at the condition media in DLBCLs. Myung is looking at this in TLLs. So we have a breadth of different heme malignancies we're looking at. And the one thing I can say again, one of the reviewers asked, is it just a adipocyte condition media, or do you see it with other condition media? So we tried condition media from osteoclasts and osteoblasts, and we don't get the same effect. So it seems to be something specific to what the adipocytes are secreting that changes the biology of these cells.
Great, thanks.
Thanks.
So Ben Miller.
So excellent talk, Curtis. I think you're in a great space with the different biologies, the metabolism, the cancer biology. I had two quick questions.
Mm-hmm.
In some of the preliminary data that you had showed, is there any gender bias? You know, in some of the biologies that we're interested in, especially in obesity, we see differences between males and female. And then my second question is, if you add soluble galectin-9 to your SCM media, can you recapitulate the adipocyte media?
So again, another reviewer. I get a chance to see all the reviewers from my papers at one time, so this is great. So for your first question, or at least let me answer your second question, which was actually a reviewer's comment that we did. So we've added recombinant Gal-9, and what we find is it doesn't have any effect. Unfortunately, we didn't add it in the stromal cell condition media, so I can't answer that. We added it to our RPMI, and we don't see any changes. And, you know, again, we don't know how closely this recombinant Gal-9 actually mimics what's shed from a leukemia cell, because proteases actually relieve Gal-9 from the surface of these cells. Regarding gender differences, we're looking at that now a little bit more closely with the data mining data that we have. So I can at least speak to the mice. It seems like the disease the kinetics of disease may change based on male or female mice that we use in these studies. So there may be an opportunity to actually go back and try to actually increase our N and do another, sex match control experiment to speak more intelligently on that question. But my short answer, and I guess my favorite hypothesis, I would say, is that I think there will be sex differences, and that may be something that we can tease out in future experiments.
All right. Okay, Jill Slansky.
Curtis, that was super satisfying. Thank you.
Thank you.
Okay, so my question is, do you know if any of these responses are can go in the other direction? Are they reversible? Like the big question for everyday life would be, like, if we get a diagnosis and then go on a diet, is that going to help? Or if anyway, I'll just
Yeah. So I got your question. So there was a really nice study published earlier this year by Steve Medelman, who is also an obesity person out at UCLA. And I think this may have been blood, but don't quote me on that. But essentially the gist of it was he had mice on high-fat diets and switched them to lean diets. And what he found was that you actually can't completely reverse the phenotype. And so there may be some type of epigenetic program. So it's just like, James and I always talk about the microenvironment and natural selection. I always argue that, a cell doesn't know how long it's going to be in that environment before it switches over. And so just like anything, there's an adaptation. So what we feel happens is that, if you're in a certain category too long, like obesity, your cells will just adjust to that microenvironment. And so even if you lose weight, some of the things are reversible, some of the things are not. Now, we've actually modeled that in our experiment. So what we did was we actually took our leukemia cells out of the ACM, put it back in normal media to see if the cells would rebound or if there was still chemosensitivity. And what you see is kind of this intermediate phenotype. So the short answer to your question is some things are reversible and some things are not. And I think there's a time dependency. So I think the longer the longer you keep these cells in these conditions, the harder it is to reverse the phenotype and get what you would consider to be a more favorable phenotype, which is what we think would happen in the lean context.
Nice, thank you.
You're welcome.
And Jen Riker? Jennifer, you're there?
I have to ask you about hormones, of course. And Ben might have been getting at this, but so androgens are higher in women with PCOS and of course higher in men, and they are known to regulate the galectins and a bunch of them. And so, and the other thing is, I've seen some studies with higher BMI affecting macrophage polarization. So I wanted to know, and skewing them towards the M2. So I wanted to know if you've started to look at macrophage polarization and function in your system.
So yeah, so we haven't looked at macrophages directly yet, but now we have the onus to. So we just got more human data back. So we did a nanostring analysis on PBMCs isolated from lean and obese patients with disease and healthy patients as well. And one of the main signatures that jumped up was like, M2 macrophage polarization and increased MDSCs. And so given now that the signature is actually jumping out of our human data, definitely, hopefully I can get some funding and can grow my lab a little bit, because I would love for someone to join and actually do some macrophage work. So there may be some opportunities for me to work with experts like you to try to figure out like what's going on. Hormones and definitely not my expertise. So my philosophy is work with smart people. And so if you have any suggestions, I'm always, my ears are always open to try to figure out, what's the best experiments to do moving forward. So
the whole systemic hormones are changed by obesity. So that would be great.
Yeah, definitely. Thank you.
Right. And last but not least, Eduardo.
Hey, Curtis. Hey, great talk, by the way. Really enjoyed it. You taught me a lot today, so thank you so much. And I love those two pictures, by the way.
Oh, thank you so much.
Nice.
Appreciate it.
Curtis, this isn't necessarily like my area of expertise, but my understanding is that there's a difference between adipose, and you have white adipose and you have brown adipose, and each of them has slightly different functions. Mm-hmm. Have you investigated how each of these different types of adipose tissues could ultimately impact either the function of the T cell or ultimately promote tumor growth?
Yes, so this is actually part of Jamie's thesis project. So he's actually doing a lot of characterization of the adipose tissue from mice from different anatomical locations. We actually try to phenotype our in vitro derived adipose tissue. And this is actually mostly brown adipose tissue in nature. They more phenotypically resemble that on a molecular level. So we're trying to do this now ex vivo from mice. We can see that there is differences in the subtypes' abilities to actually induce chemoresistance. And we also, if I can get my hands on it, I'm not good at doing IRBs, so I'm learning a lot of stuff right now as a new investigator, but I'm trying to do an IRB to work with clinicians. And we got this great idea from one of Jamie's committee members that, we have a lot of people down south getting none else than liposuction, right? And so if I could actually get, the fat that they're throwing away for research purposes, that's exactly what we're going to do. So we're hoping that we can get a large depot of fat from surgeries and then actually do experiments and then be able to speak to the primary subsets from people and how they may differentially impact disease. So hopefully more to come on that at some point.
Excellent. I'm sorry, James, I had a follow-up question. It's kind of related to Paul's earlier question. Is there a difference, is there a bias in maybe race or ethnicity with regards to how these tissues actually distribute or what their effects might be?
So yeah, so I'm actually a Trek Fellow, which is this program at Yale. I was fortunate enough to get into this program, and there's actually this pretty much everyone who works on obesity. You have the the clinicians, you have the nurses, so you get all these obesity people in the room, and then it creates this really intense and fun atmosphere, academic atmosphere. And so what I've learned is that when people who do DEXA scan, they actually are some papers that report that there is a fat distribution difference. One of the things that came out of one of my meetings was that they found that if you actually look at clinical trials and how for example, African Americans process weight and lose weight compared to everyone else. A lot of the trials concluded that, you know, for lack of not being PC, that Black people don't adhere to clinical trial protocols, which is baloney. It was incorrect because the data didn't support that. When you actually started using the correct techniques, what they found is that people of color, fat changes the muscle. Right. So the conclusions were, oh, they're not losing weight, but that's not what happened. It was conversion of fat to come more like muscle cells. And so there's actually some data that I'm really, really excited to try to partner with someone to look at differences in fat and how adipocytes from different people of different races actually behave. How do they respond to hormones or cytokines? And then what's the impact when you actually do studies in maybe humanized mice using fat from actually humans. So, you know, I have some thoughts on that based on this workshop. But yes, there are studies out there that people are looking at it, and I'm salivating to get my hands on some of this material as well. So
That's awesome. Thank you, Curtis.
thank you. Appreciate it.
This has really been fantastic. And so Curtis, I'll talk to you another time about my questions and stuff, because I wanted to give I know there are going to be people that are hanging on to this talk to have what's left of the hour to meet with you. And so for everyone else, you can sign off, but if you're planning on staying around for the lunch with Curtis, just stay on the Zoom. And otherwise, really congratulations for a great talk, Curtis. And I look forward to talking to you soon. So take care.
And James, could you do me one favor?
Yeah.
I would love to see all the comments. Could you screenshot some of the comments? So if
Oh
there's
yeah, I'll do that right now.
anything that I missed, I hopefully can email the person and try to address their comments, because I'm certain I went very close to time. So
Just
I
did it.
apologize for that. So
I got them now. And I don't know if the ones at the very top probably weren't as, relevant, but I think the ones down at the bottom I caught. The ones at the top were all about your beard.
Oh, okay. All right.
So anyway, I think I got them. I'll email them to you, and that'll serve as an impetus for us to set something up. So anyway, thanks and take care. And if you're going to stay on, stay on. And if not, you can log off. Take care.
Thank you, everyone, and happy holidays.